my name is Uzair Hashmi, and for my profile details, please visit scardia.com. Let's begin today's lecture. In today's lecture, we are going to discuss how the urine is formed by the kidneys, and we are going to discuss in detail the functions and the anatomy of the kidneys. We are going to discuss how the blood flows through the kidney and which is basically the glomerular filtrate rate, and we are going to discuss how this blood flow that is passed through the kidney is being controlled. So let's begin. Let's have a look at the lecture's outline. First of all, we are going to discuss what are the multiple functions of the kidney. We are going to discuss the physiologic anatomy of the kidney, and we are going to discuss what are the different parts of it and how these parts differ from each other in terms of their functions. We are going to discuss the physiologic anatomy of the bladder along with the kidney. We are going to discuss how the bladder is filled, and when the bladder is filled, what kind of signals they are being transferred. We are going to discuss in details about the reflex, which is known as the Micturition Reflex. We are going to discuss its abnormalities as well. We are going to also get a deep overview of the renal blood flow and the autoregulation of the glomerular filtrate rate, and along with that, the renal blood flow. So let's begin. First of all, let's begin with the multiple functions of the kidney. The kidney in our body performs quite a variety of functions, including from the excretion of the metabolic waste and the foreign chemicals towards the regulation of the water and the electrolytic balance. We are going to discuss in detail each of these points in the coming slides. This slide is just an overview of what we are going to discuss in next 3, 4, or 5 slides. We are going to discuss the atrial pressure, secretion, metabolism, and excretion of hormones, and we are going to discuss, last but not the least, the gluconeogenesis, the process of making glucose by the kidneys. Now, the first thing was the excretion of the metabolic waste products and of the foreign chemicals. So, the urea is basically formed from the metabolism of the amino acids, and the amino acids are the very basic structure of the proteins, and they are the building blocks of the proteins. Along with that, the creatine from the muscle creatine is also a part of the excretion of metabolic product. Along with that, the end product of the hemoglobin breakdown, that includes bilirubin as well. It also includes the metabolites of various other hormones. We are going to discuss in detail about these hormones as well. The other important thing is the regulation of the water and the electrolytic balance. And this is primarily the most important function of the kidney, along with various other functions which are equally important as well. This regulation of water and the electrolytic balance is because of the reason that that the kidney has to control the homeostasis. What I mean by homeostasis is, for example, let's talk about a case which varies from person to person. A person's eating and drinking habits determine the excretion rate, which of course match their intake of the various substances. So for example, if you're intaking much more substances, you are going to excrete a lot, but that is directly proportional to each, and then there is no other say in that. Let's take an example of the sodium. For example, at this particular concentration, this dotted line, you are taking sodium in the normal dose concentrations. But at a particular day, let's say the diet you took contained lots of sodium. So what happens basically is the levels of the sodium will rise immediately. This will cause, of course, because the sodium has increased suddenly, so the level of excretion of the sodium will also increase. And there will come a particular point when the sodium retention will take. This particular graph shows the level of the sodium retention. Now, this delay in the retention of the sodium is because of the reason that the body is basically acclimatizing towards the new increase in the sodium uptake. So it takes a bit longer for the body to start excreting sodium. And at this particular point over here, the body starts secreting sodium, and when the sodium level decreases, the sodium loss takes place, and this again is the retention time, because the body is excreting sodium at a very high level, so it has to make sure that the sodium level has decreased. So in order to make sure, this graph, this hyperbolic graph is formed. It also includes the regulation of the atrial pressure, which of course is maintained by the excretion of the variable amount of sodium and water that we've discussed in the previous slide. It also controls the short-term atrial pressure regulation by secreting various vasoactive factors, for example renin, which basically increases the production or stimulates the production of the angiotensin from the angiotensinogen. We are going to discuss the Renin-Angiotensin System at the last of this lecture. It also includes the regulation of the acid-base balance, because by excreting acids and by regulating the body fluids, it also causes the excretion of the sulfuric acid and the phosphoric acid that are generated as a result of metabolisms of the protein. It also includes the regulation of the erythrocyte production, and how a kidney regulates the production of the erythrocyte is because of a hormone which is known as the erythropoietin, also known as EPO. It is stimulated only in the case of the hypoxia. Now, hypoxia is a situation where the oxygen stress is being produced, and whenever the kidney will face hypoxia, it will cause the stimulation of the erythropoietin, which will ultimately go towards the bone marrow and will start the production of the red blood cells or the erythrocytes. Now, several kidney disease are in the diseases where the kidneys are removed have been replaced on the hemodialysis. This particular hormone or the erythropoietin is not being produced, which decreases the production of the red blood cells, of course, which causes the anemia to develop as a result of decreased erythropoietin production. Gluconeogenesis, also known as the glucose synthesis, is the process that occurs in the kidney. For example, whenever the glucose that has been synthesized from the amino acids and the other precursors during the prolonged fasting, this particularly function of the kidney will be taking place only in case of the prolonged fasting situations through a process known as the gluconeogenesis. Now, the chronic kidney disease or the acute failure of the kidneys have disrupted homeostasis, and as a result of it, gluconeogenesis does not occur, resulting in the various abnormalities we are going to discuss. So several abnormalities of the body volumes will occur, along with the composition will change, of course, over the period of time. Now, coming towards the physiologic anatomy of the kidney, the kidneys are present at the posterior region of the abdomen, and it is not included in the peritoneal cavity. It lies just beneath the peritoneal cavity. Now, if we look at this particular section, if we dissect the kidney from top to bottom section, not horizontally, transversely, or longitudinally, but in top to bottom section, we are going to see this particular structure. So overall, a kidney is being divided into two major portions. Number one is known as the cortex region. The outer region is called the cortex region here. The renal, abbreviated as the renal cortex. The inner region, which is present inside the dotted lines here, you can see this is known as the renal medulla, as marked here as the renal medulla. Now, the renal medulla is being divided into several forms of tissues known as renal papillae. Now, renal papillae empties themselves into the minor calyx. The minor calyx empties themselves into the major calyx, which then forms the ureter, and this particular ureter empties itself into urethra, into bladder, following the urethra. Now, this particular thing is a capsule of the kidney, which basically protects or keeps the very sensitive tissues of the kidneys intact in place. Now, the renal blood flow is basically 22% of the cardiac output. If we consider at this particular point, the renal artery enters through the ileum. I forgot to mention one particular point in the previous slide. This particular portion, this medial portion of the kidney, is known as the ileum, and it contains all the arteries, all the veins. It also contains the portion where the sympathetic and the parasympathetic systems nerves are coming. It also contains the lymphatic nervous system, lymphatic systems. So in case of the arteries, the renal arteries come in the ileum section. It is bifurcated or divided into segmental arteries. Each segmental arteries then further subdivides into interlobar arteries. These interlobar arteries then further subdivided into arcuate arteries, which further subdivides into the interlobular arteries. Now these interlobular arteries, as can be seen here, after the arcuate artery, these over here are the interlobular arteries. Now interlobular arteries over here is being bifurcated into the afferent arteriole. Now these afferent arteriole further subdivides when they move into the Bowman capsule in the glomerulus filtrate, forming the glomerular capillaries. And this is a point where the glomerular filtration is going to take place. We are going to discuss in detail about that. Following that, after the glomerular capillaries, the efferent arteriole. Efferent arterioles mean exiting arteriole, which is exiting. Results in the efferent arteriole. This particular efferent arterioles then further subdivides into the peritubular capillaries. These are the peritubular capillaries that are spread through the entire loop of Henle. These interlobular arteries then sums up to form the interlobular vein. The interlobular vein is further joined to form the arcuate vein, and the arcuate vein over here can be seen in this particular diagram. The arcuate vein then moves to form the interlobar vein, which ultimately forms the renal vein. Now this is a functional unit of the kidney, which is known as the nephron. Now it is important to consider that in a particular kidney, on average basis, approximately 1 million nephrons are present. Now the important fact about these nephrons are that the kidneys are not able to regenerate these nephrons. So whenever a particular nephron is being damaged, it's being damaged. So with the passage of time, with the passage of age, the amount or the concentrations of the nephron starts decreasing. And after the age of 40 or 45, the concentration of the nephrons starts decreasing 10% by every 10 years. Anyhow, coming back towards this particular slide, the nephron is being divided in two major sections. The upper is known as the cortex. The region above the dotted line is the cortex. The region below the dotted line is the medulla. Now, the nephron has been divided into two major portions. Number one is the glomerulus, and the other one is the capillaries, or the collecting ducts, along with the loop of Henle. Now let's discuss one by one. Now this particular thing over here is the glomerular filtrate, where the glomerular filtrate will enter through the glomerulus, and it will move from this particular tubule, which is known as the proximal tubule. This particular proximal tubule will move down, and this particular portion, this U formation, is known as the loop of Henle. It will move down towards the thin segment, which is known as descending limb, also known as descending loop of Henle, and it moves upward, forming a majorly thick section of the limb known as descending loop of Henle. Now ascending loop of Henle then moves forward to form the over this particular, this is very important region known as macula densa, which is in the proximity of the Bowman's capsule. And I'll tell you in details about the coming lectures, about the role of the macula densa, but its important function is to control the process of glomerular filtration and the autoregulation. We're going to discuss in detail how. This then ultimately forms into the connecting tubules, where it empties themselves into the cortical collecting tubules, which empties, now 8 to 10 cortical tubules then empties themselves, joins to form the medullary collecting tubules, and finally the collecting duct. Now, this particular point, I'm going to discuss two major type of nephrons. The one is known as the cortical nephron, and the other is known as the juxtamedullary nephrons. The difference in these nephrons are because of two major reasons. Number one is their location in the cortex and the medulla. So, as the name indicates, the cortical nephron, they will be present towards the proximal, the upper portion of the cortex. And they will have, of course, a very small loop of Henle, which will be just touching the medulla, the outer region of the medulla. While in case of the juxtamedullary nephron, as the name indicates, their glomeruli will be just below, just above the medulla in the cortical region, and their loop of Henle, the major portion of the nephron, will be in the medullary portion. Moving forward towards micturition. Now, what is micturition? Now, micturition is a process through which the urinary bladder empties itself when it becomes filled of urine. Now, in order to do so, there are two major steps that are involved. Number one step is, of course, in order to empty anything, it first have to be filled, right? So the bladder fills progressively, slowly and steadily with the passage of time as the blood comes through the glomerular filtrate and the fluid is being taken up, and after the reabsorption and the excretion, urine is being formed slowly and steadily, and urine moves from the ureter towards the bladder. So this is basically the short summary of progressive filling of the bladder. So the bladder will fill progressively until the tension in the walls of the bladder. The walls of the bladder are usually the smooth muscles, but they can be controlled voluntarily. We're going to discuss in detail about that as well. So the tension, when it will rise about a particular threshold, it will start sending signals. Now these signals will be sent in terms of a reflex known as the Micturition reflex. So whenever the Micturition reflex starts, we're going to discuss in detail about the properties of the Micturition reflex. The end result is the emptying of the bladder, and of course if it fails, for example, if the micturition reflex has occurred but urination has not been done, so it produces a conscious effort, sends a signal to the particular regions in the brain known as the pons, where a conscious desire to urinate is being produced. So in summary, micturition reflex is an automatic or the autonomic spinal cord reflex, but one of the important point is to be considered is it can be inhibited. This particular reflex can be inhibited or facilitated in both of the cases by the cerebral cortex, which is basically the voluntary portions of the brain. Now coming towards the second part, after discussing the functional unit nephron and the renal blood supply along with the physiologic anatomy of kidneys, we are going to discuss the physiologic anatomy and the nervous connections of the bladder in one go. So the bladder has been divided into two major portions. This particular part over here, the balloon form, is known as the body, and this particular region in the neck form is known as, of course, as the name indicates, is the neck. So this particular body is being composed of the smooth muscles, and these smooth muscles are known as the detrusor muscles. At the end of this particular body is a trigone. The urethra passes through the urogenital diaphragm layer that contains a particular type of muscles. These are the voluntary muscles. These can be seen over here. These are known as the sphincter muscles. These sphincter muscles are also present in the gastrointestinal tract as well, the stomach sphincter muscles as well. So these muscles are the voluntary muscles. The nerve supplies to the bladder is through the sacral plexus to the spinal cord. Now, if you can recall, the spinal cord has been divided into four major portions: thoracic, cervic, lumbar, and sacral region. This L stands for the lumbar, S stands for the sacrus. Now the sacral plexus to the spinal cord. Now the sympathetic systems move from the lumbar region, and the parasympathetic region moves from the sacral regions. Now, this has been divided into two major types of nerve fibers. Number one is the sensory nerve fibers. Number two is the motor nerve fibers. The sensory nerve fibers, as the name indicates, is to sense. But in terms of this particular diagram, we are talking about bladder. So what do you think it will be sensing? Yeah, it will be sensing the extent of pressure that has been developed on the walls of the bladder, on the walls of the body of the bladder, that is the smooth muscles. We have discussed the determinants. Now, when the pressure has been developed, it will start sending signal. That particular thing which we recalled or renamed as the Micturition Reflex. And the motor fibers, that includes the pudendal nerves, that includes the sphincter, external sphincter. So the basic purpose of the motor nerve fiber is to voluntarily control the opening of the urethra for the conscious urination. Now, in this particular slide, we are going to discuss Cystometrogram. Now, what that is basically, it basically tells the bladder wall tone with respect to the intravesicular pressure. The pressure peaks, okay, let's start off whenever there is a very low volume of the bladder, the pressure is almost zero. When it moves from 30 to 50, around 30 to 50 millimeters of the bladder is being filled, the pressure increases up to 5 to 10 centimeters of the waters. As it reaches to 350, you can see the gradual increase in the intravesical pressure. But as it moves from 350 onwards, there you can see a rapid increase in the intravesicular pressures. Now, these peaks, these dotted lines, these are basically the micturition waves that you can see in the cystometrogram. And these are caused by the micturition reflex, because it's a self-generating reflex. When it starts, it keeps on moving until the bladder has been emptied. So it will, with the passage of time, progressively will keep sending signal that the bladder has been filled and there is a need to remove. So the micturition reflex is basically a result of a stretch reflex. We have discussed that particular stretch reflex in the previous slides, which is initiated by the sensory stretch receptors. We have discussed that as well in the bladder wall, of course. Now, the bladder filling is directly proportional to the reflex occurrence and the muscle contraction. Now, there are two things. Number one, the bladder filling, the particular thing, when the bladder is being filled, is directly proportional to the reflex occurrence. For example, if the bladder has been filled to 50 or 60%, when the percentage of the bladder that is to be filled is increasing, the occurrence of the reflex action will increase, of course. It will continuously send signal to the brain that for the urge to stimulate for the urination, and also the muscle contraction, the sphincter muscle contraction and the smooth muscle contraction. Of course, it is self-regenerative, and at that particular point, when the bladder is starting to get emptied, the self-regenerative reflex, which I have discussed, that begins to fatigue. Now, this particular regenerative cycle of the micturition reflex ceases at that particular point. When it starts fatiguing, the regenerative cycle starts to cease. And after that particular time, the contraction that the bladder feels will stop, and the bladder will start relaxing. Now, the micturition reflex is a single complete cycle. If we divide into three major phases, number one is the progressive and the rapid increase of the pressure, and this particular pressure is felt by the bladder wall. Number two, pressure has been increased to up to a particular point. This will cause the sustainability of a pressure, a period where the pressure will sustain. Number three would be the retention, the return of the pressure to the basal tone of the bladder, and this will occur only when the bladder is being empty after the process of urination or the micturition. So, facilitation or inhibition of the micturition by the brain. Now, since the process of the micturition is usually an autonomic spinal cord reflex system, but it can be controlled by brain. And when I say brain, I'm talking about two major regions, two major centers in the brain. The strong facilitative or the inhibitory centers in the brain cells, they are located mainly in the palms. Also, there are several centers that are located in the cerebral cortex, and that are mainly inhibitory, but they can become excitatory. We are going to discuss in detail about the details of the two, how they are inhibitory, and in what particular scenarios they can become excitatory. Now, the higher centers that normally exert final cohort of the micturations are as follows. Number one is the higher center, of course. It will keep the micturition reflex partially inhibited when it starts, of course, because the conscious system or the voluntary system of the brain wants the bladder to be filled completely after it sends signals, after it produces the conscious effort to urinate. So at that particular point, when the bladder is one-fifth filled or two-fifth filled, or there is a capacity for the bladder to be filled, that particular high centers will keep the micturition reflex partially inhibited. This will be followed by the higher centers that can prevent the micturations by contraction of the external bladder sphincter. I have discussed in detail that the sphincter muscles are controlled by the parasympathetic systems from the sacral portions. These higher centers have the ability to control the sphincter muscles as well for the process of the conscious urination or the conscious micturition. So what basically happens in when the bladder has been particularly filled, 3/4 or completely filled, or when it's time to urinate, the cortical centers starts to facilitate at that particular point when the bladder has been filled to help initiate a micturition reflex. That particularly partially inhibited micturition reflex will that particular inhibition by the higher center will be reduced at that particular time, and at the same time it will inhibit the external urinary sphincter muscles. So up till now we have discussed what are the multiple functions of the kidneys. We have discussed the physiologic anatomy of the kidneys. We have discussed the structure and the function unit of the kidney, which is the nephron. We have discussed the urinary bladder as well, the physiologic anatomy of the bladder. We have discussed the process of micturition. We have also discussed how the transport of the urine occurs from the kidney, from the nephron towards the minor calyx, towards the major calyx, towards the ureter, and from ureter moving towards the bladder, and from bladder to urethra. We have discussed the micturition reflexes as well. Now we are going to discuss about the abnormalities of the micturition. Now because it's an autonomic reflex system, so any injury or any abnormalities to the spinal cord can result in the abnormalities of the micturition if that particular portion of the autonomic nervous system or that portion of the spinal cord is being affected. Number one is the tonic bladder. It is caused by of course, destruction of the sensory nerve fibers. Now, when the sensory nerve fibers will be destructed, there will be no stretch signals from the bladder, and the control on the bladder will be lost. Now, there are many different causes for the tonic bladder. Number one, of course, is the bladder control loss that can occur because of the variety of reasons. That includes overflow incontinence. It can include because of the crush injury, because the injury can disrupt, can hit or damage the spinal cord, and it can damage the sensory nerve fibers, which causes the contraction of the urinary bladder. It also it can also occur because of several diseases, also known as, for example, in this particular case, syphilis. The tabes dorsalis is basically the damage to the dorsal root fibers, and these dorsal root fibers, they enter to the spinal cord. And in the case of the syphilis, these particular dorsal root fibers, they are being damaged, which produces this particular condition known as atonic bladder. The automatic bladder caused by the spinal cord damage above the sacral region. Now, sacral region I have discussed in detail in the bladder slide as well. When this particular portion is being damaged, so the brain will have no control over this particular region. So the micturition reflexes will be of course suppressed because of the state of the spinal shock. However, many of the patients in this particular scenario can still control the urination, and how they can do so? They can do so by stimulating the skin, or either by scratching or tickling in the genital region, which basically elicits a micturition reflex. The last is the uninhibited neurogenic bladder caused by the lack of inhibitory signals from the brain. This occurs because of the partial damage in the spinal cord. This partial damage which occurs in the spinal cord interrupts most of the inhibitory signals. When the inhibitory signals are being inhibited, the facilitated signals will be of course generated, and they will be excitable then after that particular time when the inhibitory signals are being released or interrupted. When the excitatory signals are being given, the facilitative impulses will go towards the bladder. This will cause, even if the small quantity of urine that is being present in the bladder, which will of course produce the micturition reflex. This micturition reflex, the function was being partially inhibited by the high centers. Now, when the high centers of the brain will lose control, what will happen is basically whenever the micturition reflex is being produced, the bladder will start urinating. This will result in the production of the frequent urination or the uncontrollable micturition reflex. Now, talking about the urine formation, it can be divided into four major sections. Number one is the filtration. This filtration basically occurs in the glomerular filtrate portion. This here is the Bowman's capsule, and this here is the glomerular filtrate. This is followed by the reabsorption. The reabsorption occurs in the tubules. This is followed by the secretion. The secretion, the secretion occurs from the peritubular capillaries that usually surrounds the loop family. And of course, number four, the excretion of urine. So excretion is basically the filtration. When you minus filtration from the reabsorption and you will add the secretion from, of course, the peritubular capillaries, you will get the excretion. Now, there are four scenarios discussed over here. And these substances are the hypothetical substances. I'm going to discuss in detail about each of these steps. Now, there is a particular case that a substance A is only filtered from the glomerular filtrate and is not reabsorbed back to the tubules, collecting tubules, or secreted back. So it will directly go towards the urine. And this particular substance A can be anything. One of the example of the substance A is the creatinine, which is produced by the muscle creatine. This particular B is filtration and the partial reabsorption. The substance B will move from the glomerular filtrate into the Bowman's capsule. It will move down towards the conducting tubules, collecting tubules, and is partially reabsorbed and partially excreted in the urine. And these includes the electrolytes. The partial reabsorption depends upon the levels of the electrolytes that are present in the body. Number C is the filtration or the complete reabsorption. So basically what happens is this particular substance C, of course, is present in the blood, and it passes from the glomerular filtrate into the Bowman's capsule. But this substance C is of very prime importance to the body. That is why, of course, it is completely reabsorbed back. And you can guess the substance C, which is a very important component that can be utilized by the body, includes the amino acids, which will of course be used in the formation of proteins and the glucose. Number 4 is the filtration and the secretion. So basically what is happening here is, from the glomerular filtrate, this particular substance is being filtered out and after filtering out, it will move to the Bowman's capsule and it will not be reabsorbed back. But because the peritubular capillaries over here are also in contact with the collecting tubules and in contact with the loop of Henle. So basically what will happen is the remaining amount which was not secreted into the Bowman's capsule will also be secreted into the urine, just to make sure that their levels in the blood remains to the very minimum. So these includes the organic acids and bases. So talking about the glomerular filtration, the first step in the urine formation. So GFR stands for, of course, glomerular filtration rate, GFR. It's basically the 20% of the renal plasma flow. So that's, you can say, formula for the GFR. So the glomerular capillary membrane, if you're going to discuss in details about the glomerular capillary membrane, we can see over here that is composed of three major portions. Number one is the endothelium of the capillary. You can see over here the endothelium which contains the fenestrations. I'm going to discuss in detail about that as well. This also includes a basement membrane that basically separates the endothelium from the epithelium. Now, this layer of the epithelium contains very important cells. These are known as the podocytes, and these podocytes are responsible for the transfer of the glomerular filtrate from the blood into the Bowman's capsule. Now, two important points are to be considered over here, that the filterability of the solute is inversely related to their size. What I mean by saying this is that the substances which have larger molecular mass, which are larger in size, they are very less prone to be filtered out because of their size, of course. And small size bodies, they can be easily filtered out. That is why amino acids can be filtered out at that particular time, although they are completely reabsorbed back in the stage 3, which is the reabsorption that we discussed in the previous slide. The negatively charged molecules are less filtered than the positively charged filter molecules. I'm going to discuss its reason in the coming lectures. Added that they have the equal molecular size. So if you have two particular things with same molecular size, molecule A and molecule B, they are of same size, but molecule A has positive charge and molecule B have negative charge. So basically what will happen is molecule B will be less likely to be filtered as compared to molecule A. So what are the determinants of GFR, or the glomerular filtration rate? Now there are four determinants that you should be keeping in mind, and this is very important slide to consider. Number one is the hydrostatic pressure. And the hydrostatic pressure is the pressure that is present inside the glomerular capillaries. So it's the pressure exerted by the blood, by the glomerular capillaries towards the Bowman capsule, so that with this particular pressure development, the filtrate can enter through the podocytes into the Bowman's capsule, and this pressure is usually 60 millimeter per mercury. Now, this particular pressure, the glomerular hydrostatic pressure, will of course promote the filtration. The greater the glomerular hydrostatic pressure will be, the greater will be the process of filtration. Number two is the hydrostatic pressure of the Bowman's capsule. Because the Bowman's capsule is present against the glomerular capillaries, so if you will develop the pressure towards the glomerular filtrate, the Bowman's capsule, what will happen basically is the rate of the filtration will reduce, because if you increase the pressure rate, if the glomerular capillaries are pressing in this particular direction and the hydrostatic pressure of the Bowman's capsule is in this particular direction, so it will basically be opposing the glomerular filtrate to enter into the Bowman's capsule. So in order for this particular thing to go there, this particular pressure has to be less. And this pressure is shown over here as the 32 millimeter mercury. Third and the most important thing is the colloid osmotic pressure of the glomeruli capillary plasma proteins. Now these are the proteins that are present in the glomerular capillaries, and these of course opposes the filtration. In opposition to the plasma proteins that are present in the glomerular capillaries, the proteins that are present in the Bowman's capsule will of course promote the filtration. So the net filtration will become the hydrostatic pressure of the glomerular filtrate. You will minus the hydrostatic pressure of the Bowman's capsule in order to get the net effect. And of course, you will deduct the glomerular oncotic pressure, which is of course 32 millimeters per mercury. Kf here represents a particular constant. Now, moving towards the physiologic control of the glomerular filtration and the renal blood flow. Now, there are various factors that control the glomerular filtration rate and of course resulting in the renal blood flow. Number one is the sympathetic nervous system. When the sympathetic nervous system will be activated, of course, it will decrease the glomerular filtration rate. Now, there are certain hormones, including norepinephrine, epinephrine, endothelin. They will function to basically in the vasoconstriction. Now, vasoconstriction is basically the constriction of the blood vessels, and by constriction means it will reduce the volume of or the diameter of the blood vessels. And when the blood diameter will be reduced, the flow of the blood will be reduced, and hence the glomerular filtration rate will be reduced. The angiotensin II system. We are going to discuss in detail in the coming slide what is actually the function of the angiotensin II. It will of course causes the constriction of the efferent arterioles that are basically present after the glomerular capillaries. So basically we have efferent arterioles moving into the glomerular capillaries, and the glomerular capillaries, when moving out, they form the efferent afferent arterioles. There are also an endothelial derived nitric oxide. Now this nitric oxide functions as the vasodilator. Now, by saying the vasodilator, it will increase the diameter of the blood vessels in contrast to the function of the norepinephrine, epinephrine, and the endothelium. Of course, it also include the prostaglandins and the bradykinins that tends to increase the GFR. Now let's talk about the autoregulation of the glomerular filtration rate. We have the feedback mechanisms which are intrinsic to the kidney. Now these feedback mechanisms we are going to discuss in detail are present inside the kidney. They are intrinsic to kidney, innately present. And the purpose of these feedback mechanisms are to keep the renal blood flow, number one, and the glomerular filtration rate, number two, constant. These feedback mechanisms will be basically monitoring the renal blood flow, number one, and the glomerular filtration rate, and they will keeping these two things to a particular level. Despite the pressure has been increased, despite the pressure has been reduced, whatever the condition is with the atrial pressure, if the feedback mechanism or the autoregulation of the glomerular filtration rate, this particular mechanism which is intrinsic to kidney is live, there will be minimum alteration in the renal blood flow and the glomerular filtration rate. Now, this particular range is about 70 to 160 millimeter per mercury. Now, let's think of a scenario where this autoregulation is not present. Now, whenever this particular autoregulation is not present, even a very slight increase in the blood pressure, it will drastically change and produces significant change in the glomerular filtration rate. Let's say, for example, we have 100 millimeter mercury blood pressure. At this particular pressure, the glomerular filtration rate, GFR, is 180 liters per day. The GFR is 180 liters per day. That produces 1 liter of urine per day at the pressure of 100 millimeter mercury. Now, since we have assumed that the autoregulation is not present, so let's increase this particular pressure to 25%. And after increasing it to 25%, what will it become? It will become 125 millimeter mercury. So it has increased to 25 millimeter per mercury. So this will increase the glomerular filtration rate from 180 liters per day to 225 liters per day of the glomerular filtration rate. And over there, when the pressure was 100 millimeter mercury, 1 liter of the urine was produced, here 46 liter of urine will be produced. So that is how the production of the urine and the glomerular filtration rate is altered if this particular autoregulation system is not present or is not active, these particular feedback mechanism. So this particular renal autoregulation is being divided into two major portions. At this particular hand we have myogenic mechanism, at this particular hand we have tubulo-glomerular feedback. And there are other factors, of course, as well. We cannot negate them, but these are of minor importance as compared to these two mechanisms. That includes the neural, hormonal, or the vasoactive substances that are basically the extensions of these two autoregulation systems. Now, what happens in the myogenic mechanism? The atrial smooth muscle contracts. Number one is the contraction of the atrial smooth muscles, and it will, of course, causes the increase and the decrease in the vascular wall tension. So basically what happens is atrial smooth muscle has been contracted, and this will and of course it will cause the relaxation in response to increase and decrease in the vascular wall tension. Now this particular myogenic system, it will contribute towards the 50% of the total autoregulatory response, number one, of course, and this particular myogenic mechanism starts and it reaches to its particular peak within a response time of 3 to 10 seconds. So it has a response of 3 to 10 seconds. It is a property of pre-glomerular resistant vessels. These myogenic mechanisms are present in various vessels. We have discussed the physiologic anatomy of the kidneys, so you must be aware of the capillaries or the blood vessels that are passing throughout the kidneys. For example, arcuate capillaries, arcuate vessels, interlobular, and that of the afferent atrioles. Now this particular myogenic mechanism is not seen in the efferent atrioles, and there are many different reasons for that. At this particular point, only one important difference is to be considered, and keep in mind that this particular difference, that the myogenic mechanism is not present in the efferent atriole, is because of lack of the voltage gated calcium channels. Now why the myogenic mechanism is not present in the efferent atriole, it's a whole another debate that we are going to discuss in the second portion of this particular lecture. So this slide basically tells the summary of the myogenic autoregulation. Number one is when the atrial blood pressure increases, what will happen? The afferent atriolar blood pressure will of course increase. This will produce the stretch in the atrial walls. Now this stretch will be sensed by the myogenic stress receptors. These myogenic stress receptors will cause the opening of the voltage gated calcium channels. When the voltage gated calcium channels will be opened, this will cause the influx of the calcium from the extracellular fluid, ECF, from the extracellular fluid to the vascular smooth muscle cells. Now what will happen when, if you can remember, the calcium that enters from the extracellular fluid to the muscle cells will bind to the troponin head. It will cause the contraction of the muscles. When the muscle will contract, this will cause the vasoconstriction, the constriction of the muscles of the blood vessels. Now after the vasoconstriction has been occurred, so basically what will happen? It will minimize the changes in the afferent atriolar blood flow. So because the atrial blood pressure has increased, this would have increased the glomerular filtration rate, but that particular rate had to be stabilized. So what happened was there was the influx in the extracellular fluid, which causes the vasoconstriction, and that particular pressure decreases. That particular atrial pressure, which was initially increased, was decreased, and the changes to the afferent atriolar blood flow was minimized, and this ultimately minimized the changes in the glomerular filtration rate. Number two is the mechanism of the tubulo-glomerular feedback. Now this is a feedback mechanism very important that links sodium and chloride concentration to the macula densa. I have discussed in the very beginning of this particular lecture about the macula densa that is present in the proximity of the Bowman capsule. Now we are talking about that particular macula densa. Now this particular macula densa with the control of the renal arteriolar resistance. So what basically happens in this particular scenario is this particular tubular glomerular feedback mechanism acts to the acute perturbations, the disturbances that are very rapid, that has been raised, that are not chronic, that are very acute, that have not developed with the passage of time, with the progression of time, but that are very new and they are very severe in the delivery of fluids and the solute to the JGA, which are the juxtamedullary glomerular apparatus. These are the type of cells that are present in the juxtamedullary nephrons. So it has two components. Number one is the afferent atriolar feedback. Number two is the afferent atriolar feedback. Of course, that is hormonal because of the absence of the voltage-gated potassium channels. That has been discussed in the previous slide. It also helps, number one, is in the autoregulation of the GFR. It controls, of course, the distal solute delivery. The solute delivery at the distal portion of the tubules, and hence, of course, it will be affecting the tubular reabsorption, the number two step after the filtration, reabsorption, excretion. The neural regulation of the GFR includes we have discussed in detail how the sympathetic system controls the glomerular filtration rate, and that is because the sympathetic systems have nerves that innervate both the afferent and the efferent atrioles. Normally what happens is basically the sympathetic stimulation is low. In case of low sympathetic stimulation, it has less or very no effect on the glomerular filtration rate. But what happens when the sympathetic stimulation is excessive? Usually it will happen, normally it doesn't happen, but it will happen in the case of shock or whenever there is a case of ischemia or severe hemorrhage, basically in the shock situations. This will last from a few minutes to a few hours, and it will cause the stimulation of the renal vessels. And by stimulation, I mean it will cause, of course, the vasoconstriction, which will result in the conversations of the blood volume. Now, this is very important. The vasoconstriction means that the blood vessels are being constricted. Now, when blood vessels will be constricted, the volume of the blood passing through these particular vessels will be reduced. And in hemorrhage, what basically happens is the bleeding. So the bleeding, there will be a very minimum loss of the blood. So this particular neural regulation of the GFR is very important if this particular point is to be considered. Now, talking about the parasympathetic nervous system. The parasympathetic nervous system contains acetylcholine, of course, which causes the release of the nitric oxide. I have discussed the nitric oxide as a vasodilator in the previous slides as well. The nitric oxide from the endothelial cells. Now, these endothelial cells, of course, will cause the vasodilation, which will increase ultimately the GFR after the body has recovered from the shock or has been recovered from the severe hemorrhage or the brain ischemia. Now, in summary, let's talk about the hormone regulation of the GFR. Let's start with the vasoconstriction. So we have at one hand vasoconstriction that will, of course, cause the decrease in the GFR, and the vasodilator that will increase the GFR. By increasing and decreasing the diameter of the blood vessels. Now, vasoconstrictions, these includes the norepinephrine and the epinephrine. They are basically released in the stressful situation, and when they are released along with the sympathetic stimulation, the stimulation from the sympathetic nervous system. The next thing is the endothelin. Endothelin is being released in variety of conditions. Starting from the acute renal failure known as ARF, toxemia of the pregnancy. It also is being released in the vascular injury and in the chronic uremia. Number two is the other thing is the angiotensin 2. Now, the angiotensin 2 is being produced by the Renin-Angiotensin System, and they are released by the juxtamedullary glomerular apparatus cells, JGA cells. And there are also leukotrienes. And these are the two examples of the specifics of the leukotrienes, that includes LTC4 and LTD4. We are going to discuss in details about the hormonal parts of this particular course. It also includes the vasodilators. The vasodilators includes the nitric oxide that has been released from the endothelial cells. It also includes the prostaglandins. It also includes that includes the E2 and I2. It also includes the bradykinin and the leukotriene LTB4. Now let's have a brief overview of the renin-angiotensin system. The renin-angiotensin system is activated or gets live in two conditions. Either the sodium level is being low or the renal blood flow is low. So what will it cause? It will cause the upregulation of the juxtamedullary operators of the kidneys, where the receptors will be controlling the amount. Now what will happen? It will cause the production or the release of the renin from the JGA cells that I have discussed in the previous slide. This renin will convert the inactive angiotensinogen over here to angiotensin 1. This angiotensin 1 is converted in the lungs through the converting enzymes into angiotensin 2. Angiotensin 2 is then converted into the angiotensin 3. And these angiotensin 2 and angiotensin 3 are they are very powerful. Vasoconstriction causes the constriction of the blood vessels in the time span of seconds. Now, angiotensin 2 in the adrenal cortex will convert corticosterone into the aldosterone. Now, this aldosterone plays a very, very vital role because aldosterone is the main regulator of the sodium retention. Because when the level of the aldosterone will increase, what will it cause? It will cause the reabsorption of the sodium from the collecting tubules to occur. Now, this again explains the macular densa feedback mechanism, or the mechanism that is shown by the tubular glomerular feedback. That the macular densa feedback mechanism is a part of this particular thing. Now, what happens in this particular macular densa feedback mechanism? It's similar to the feedback mechanism that we have discussed in the previous slides, is that the atrial pressure will decrease. This decrease in the arterial pressure will of course cause the reduction in the glomerular hydrostatic pressure. When the glomerular hydrostatic pressure will decrease, of course the opposition from the hydrostatic pressure of the Bowman's capsule has been increased, so the net difference will be reduced. This will cause the reduction in the glomerular filtration rate. Because the glomerular filtration rate is being reduced, this is being sensed by the macular densa that is present in the very close proximity of the Bowman's capsule. We have discussed in the physiologic anatomy slide of the kidney. This macular densa will cause the sodium chloride to increase in concentration through the reabsorption of the sodium chloride. It will also cause the upregulation of the renin. The renin will be released from the juxtamedullary cortical nephrons. These renin will cause the production of the angiotensinogen 2. This angiotensinogen will, of course, be activated by the renin into angiotensin 2, which is, of course, is the powerful vasoconstrictor. It will cause the, of course, when I say that it will cause the vasoconstriction, what will it be doing? It will be basically reducing or increasing the resistance from the afferent arterioles. The resistance from the afferent arterioles, the arterioles that are exiting from the glomerular capillaries will be increased. And it will decrease the resistance from, of course, the afferent arteriole. So basically it will cause the overflow of the arterial pressure. And these two things will negatively regulate the glomerular hydrostatic pressure. For example, the decrease in this particular thing will do the opposite of the glomerular hydrostatic pressure. So if it was being decreased over here, whenever the afferent arteriolar resistance is decreased and the efferent arteriolar resistance is increased, it will negatively regulate, causing the increase in the glomerular hydrostatic pressure. So when this particular hydrostatic pressure is increased, the whole of these particular thing will be stopped, because these have negatively regulated, and the renin which has been released from the macular densa portion or the from the juxtamedullary portion to activate the angiotensin to cause the vasoconstriction. That process will be completed, and hence the feedback mechanism will be completed. Now, in this particular scenario, in this particular lecture, we have gone through a whole lot of things. Starting from the multiple functions of the kidney, we have discussed how kidneys can produce erythropoietin to regulate the production of the erythrocyte, how they can produce the glucose in the gluconeogenesis during the fasting situations. We have discussed the physiologic anatomy of the kidney. In that, we have discussed a functional unit of the kidney, nephron. We have discussed how many nephrons are present and how each particular collecting tubules can collect the urine from how many nephrons. We have discussed the micturition process. We have discussed the physiologic anatomy of the bladder along with the sympathetic and the parasympathetic system that controls the bladder. We have discussed how the urine is being transported from the kidney through the ureter into the bladder and from bladder through urethra. We have discussed the micturition reflexes and how the micturition reflexes is being regulated, and what are the abnormalities of the micturition, which one of that was the atonic bladder, which results from the injury into the sacral region. We also discuss about the glomerular filtration, which is of course the first step in the urine formation. We discussed about after the glomerular filtration, we discussed about the reabsorption and the secretion part leading to the excretion part. We discussed the determinants of the GFR. We discussed the 4 determinants of how hydrostatic pressure of the glomerular filtrate, hydrostatic pressure of the Bowman's capsule, colloid hydrostatic pressure of the proteins, plasma proteins in the glomerular capsule, glomerular capillaries, and the plasma proteins that are present in the Bowman's capsule collectively affect the glomerular filtration rate. We discussed the renal blood flow, how it is being flowed from the renal artery into interlobar arteries, from interlobar arteries into various different capillaries reaching to the afferent and efferent arterioles. Finally, we discussed how the control of the glomerular filtration and the renal blood flow occurs physiologically, and we have discussed various vasodilators and the vasoconstrictors. We have discussed the autoregulation of the GFR and the renal blood flow. And in this particular autoregulation, we have discussed how these process of the autoregulation, discussing 2 myogenic feedback mechanism, macular densa feedback mechanism. We have discussed the Renin-Angiotensin feedback mechanism, how all of these together tackle the regulation of the glomerular filtration rate along with the renal blood flow, so that the normal functions of the kidneys are being restored, and the 4 processes starting from the filtration, moving towards the reabsorption, and from reabsorption to secretion, and from secretion to ultimately excretion of the urine occurs in a very normal and positive way. That is all for this particular lecture. Thank you for watching, and keep watching Scardia.com.